Okay, so this is the third part of the series and hopefully the final one. So hopefully we can get through the entire thing without needing another break. So I'd like to continue this paper, which is called Iron Regulatory Protein, Iron Response Element Signaling Pathway in Human Neurodegenerative Diseases. So to recap very quickly, know that in your cells have these proteins called iron regulatory proteins or IRPs, and also they can sometimes be called iron response proteins. Regardless, these iron response proteins are able to interact with the iron response elements, which are secondary structures that are on the mRNA of a lot of the mRNAs that are important in iron metabolism. So genes that are transcribed from DNA into RNA that have these iron response elements on them, they will form these little shapes which can be used to modulate iron activity using the iron ions interacting with the iron regulatory proteins. And also, there might be some other iron response elements that can bind directly to iron itself, and maybe some other proteins as well, but that is besides the point. So we previously gone over some of the proteins that are associated with Alzheimer's disease and Parkinson's disease are have these iron regulatory elements associated with some of the proteins that will precipitate out of solution if their concentrations get too high. So if you have proteins that are highly upregulated in the presence of iron or in the dysregulation of iron to the point where you have way too much of it in your system, then you produce so much of this protein that it can end up resulting or make it more easy for you to have these proteins fall out of solution or precipitate out of solution, and then they can form aggregates, and these aggregates can interfere with the cell's normal ability to function and sometimes can result in cell death. So, for example, the amyloid beta plaques are formed from the amyloid beta peptides, which come from the amyloid precursor protein. And if you have too much amyloid precursor protein, you'll probably have more amyloid beta peptides that are synthesized from the APP. So as a result, if you have too much APP in your system, which can be upregulated from iron, it can result in excess or the overproduction of amyloid beta, which can result in those plaques, which can kill your cells or kill your nerve cells. So the issue with that is, I guess the double-edged sword is the APP is probably playing an important protective role in your brain to prevent the oxidative damage of iron and protecting your cells from the dangers of iron. But if you have too much of it and if it becomes dysregulated, then it's no longer, it's not just playing a beneficial role, it will also be playing a negative role by precipitating out a solution. So with that, we can move from Alzheimer's and Parkinson's and move to other human disorders that are not necessarily neurological. Anyways, although none of the point mutations in IRP1 or IRP2, and remember these are the iron response proteins, and remember you have two different types of iron response proteins in your cells. Anyways, none of the point mutations have been confirmed to cause human diseases. Some preliminary studies have implicated the potential association of single nucleotide polymorphisms or mutations of IRPs with human disease. So they haven't proven, we haven't confirmed any of these associations, but we have preliminary studies that have implicated a possible association with single nucleotide, or a single nucleotide polymorphism is a mutation, but it's a mutation of only one of the nucleotides of the DNA. So remember, your DNA has letters that they are the base pairs, right? Or they are the bases of the DNA. You have four of them normally in DNA. What happens is if you have a single nucleotide polymorphism, one of those nucleotides has mutated into a different one. And this might not necessarily even change the amino acid that it encodes for, but it can change it. That single mutation might have some sort of regulatory change on the system as a whole. So we haven't proven that any one of these mutations has caused an issue, but we are seeing some associations with diseases. So, a genome-wide association study, or a GWAS, identified three IRP2 single nucleotide polymorphisms associated with patients with chronic obstructive pulmonary disease, or COPD. So it is possible that COPD is associated with a single mutation of IRP2. Or I should say, there are three different mutations associated with IRP2 that are associated with COPD. Anyways, the levels of IRP2 protein and mRNA have been found to be higher in lung tissue samples from COPD patients than from controls. So the levels of the RP2 protein and the mRNA seems to be in higher concentrations of people with COPD. These findings indicated that RP2 is a COPD susceptible gene, and further studies are needed to establish the potential pathological role for IRP2 in COPD. So the things that could theoretically cause this increase in the mRNA of IRP2, let's just say it mutates in a way that it might possibly mutate in a way that has it be expressed more easily. So perhaps there's a mutation in the promoter region of the protein of the mRNA, which allows it to be read by ribosomes to produce the protein much more readily. There could be a mutation that will remove one of the suppressors. Yeah, there's a lot of different things that a single point mutation could do to affect the relative concentrations of a particular protein. But just so you know, if you have too much, or if you have a lot of this IRP2, this IRP2 is going to do twofold, or have two different consequences. One of them is for all of the mRNAs that have these iron response elements on them, there will be more of these iron response elements that can be bound by the iron regulatory proteins. So if these iron regulatory proteins, if you have a lot of them, even if you have some iron or a lot of iron in your system, you still might have a lot left over to still be binding. And for the in the case of the 5' iron response elements, you might have a lot of inhibition of genes that you do not want to have, that you do not want to be inhibited, or that you do not want to see be inhibited in the presence of iron, but it still isn't inhibited because you have way too much IRP2. Or in this case, you could have so much IRP2 that even if you have iron, normally you want iron to destroy these mRNAs, or you would want the high concentration of iron to result in the destruction of these mRNAs and therefore stop the protein production. But if you have so many RP2s, you could still have, theoretically, RP2s binding to these iron response elements and protecting these 3' iron response elements. And by protecting these 3' iron response elements, you are producing more protein than you want to produce. So that's theoretically one of the ways that an upregulation of IRP2 could theoretically cause pathology. Anyways, let's go back down. So mutations in the Ire motif, so the iron response element motif, have been identified in other human disorders beyond human neurodegenerative diseases. So multiple point mutations in the iron response element motif of L-ferritin, and remember, L-ferritin, so this is the iron response element in L-ferritin, and L-ferritin is a protein that is going to help store iron in the cells of your body. And I wonder if L-ferritin is explicitly for the liver. You know what? Let's look this up. L-ferritin. So yeah, so it means the light chain. So in vertebrates, the subunits are two types, the light and heavy chain, which have apparent molecular masses of 19 and 21 kilodaltons. So, okay, what just happened? Okay, so it looks like they're just the two subunits, the light chain and the heavy chain for, so it's a heteropolymer. Yeah, I'm not going to go too much into the detail of the structure of, what's it called? I'm not going to go into too much detail of the structure of ferritin. Just know that the Ire motif of the ferritin light chain, this has been linked to hyperferritinemia cataract syndrome, or HHCH, with hyperferritinemia and autosomal dominant congenital cataract. So what is this hyperferritinemia? So emia means that it is in the blood, if I'm remembering correctly. So let's look up Wikipedia, because I don't want to. Oh, we could go to iWiki. Load, please. Yeah, my internet's not been too great these past few hours, so well, my internet's just not working at all. So we will come back to that when my internet starts deciding to behave. So hyperferritinemia. So hyper meaning that there's a lot of it, and ferritin meaning there's a lot of ferritin, and emia, there's a lot of ferritin in the blood. And hyperferritinemia cataract syndrome would be cataracts that are formed by having way too much ferritin in the blood. So, and that's if I'm translating that properly. If this decides to load, then maybe we will get a better idea of what's going on. Anyways, the serum. We are. Yeah, here. With hyperferritinemia and autosomal dominant congenital cataract. So the disease is characterized by early onset bilateral nuclear cataracts and upregulated levels of serum ferritin. So, however, HHCH patients demonstrate no hematological or biochemical abnormalities in iron metabolism. So they don't appear to have biochemical abnormalities in iron metabolism, but there is a problem that is coming up, or a pathology that's coming up. Anyways, the serum iron levels and transferrin saturations of HHCH patients are normal to low. So the iron that is in the blood, the iron that you can see in the blood, seems to be normal or low. Anyways, the level of body iron is also not increased. Therefore, excluding iron overload as a pathogenic cause of HHCH, studies have demonstrated that the point mutation of the iron response element motif of L-ferritin mRNA disturbs the IRP interaction of the highly conserved IRE stem loop. So there is a mutation that happens here that destabilizes this somehow. And it influences its ability to interact with the iron regulatory protein. And so remember, this is an iron regulatory protein that is in the 5' area. So this would normally be inhibiting, this would inhibit the translation of the ferritin light chain. So normally it would be this situation here, where the presence of the iron response protein is with this iron response element. This is going to inhibit the ferritin light chain from being produced. Now, if you have a lot of iron in your system, then the iron is going to bind to the iron response protein, or iron regulatory protein, and then that is going to result in the production of your ferritin light chain. Now, imagine you have a mutation in this iron response element, which means that this iron regulatory iron regulatory protein is not able to bind this iron response element. Then what happens? Well, even if you do not have this iron here, this iron response protein is not going to be able to bind to this mutated, or this single nucleotide polymorphism version of this iron response element. And as a result, we are producing way too much ferritin light chain for our conditions. So what ends up happening as a result of that? So furthermore, the disease relevant point mutations in the Ire motif of L-ferritin lead to the abolishment of the binding of IRPs to the L-ferritin transcript, resulting in significantly increased synthesis of L-ferritin in cells from HHCH patients, which is in agreement with the upregulated serum ferritin levels observed in these patients. So we see it in their blood that they are producing a lot more ferritin than they normally would. Okay, so the point mutation of the iron response element of L-ferritin mRNA induces hyperferritinemia, so having way too much ferritin in your blood, and this may lead to the accumulation and aggregation of L-ferritin in the lens and early onset of cataracts. So what is happening here? What is happening is the ferritin is going to be upregulated so much that its concentrations are high enough that it can start to precipitate out of solution, and it precipitates in aggregates. And it's presumably, I would presume that it's precipitating with iron associated with it because it's ferritin. So ferritin is going to suck up a lot of the iron in the surrounding areas. So basically, you're having an accumulation and aggregation of L-ferritin in the lens. So, and that is causing cataracts. So furthermore, mutations in the Ire motif of H-ferritin, which is the heavy chain of ferritin, this gene may also be relevant to human diseases. So a point mutation from the A in the 49 position to a U in the 5' UTR-IRE motif. And remember, A and U are both, they're both nucleotides. If you have that change from A to U in the 5' untranslated region IRE motif of the human heavy ferritin gene, this has been associated in members of a Japanese family with dominantly inherited iron overload. So these people have a point mutation, and it was a dominant gene. So if you had either one of these, in your chromosomes, or how to phrase that, if either copy of this gene was this mutation, then you end up getting this disease, and the disease was what was it called? Iron overload. So you're basically at a very high risk of getting way too much iron in your body if you have this disease. So this point mutation in the IRE motif of H-ferritin increases its affinity for the IRP binding. So you have the opposite effect happening. So this point mutation of the IRE motif of the H-ferritin, this is increasing its affinity for IRP binding. This is leading to the suppression of heavy chain ferritin synthesis and increase in iron uptake, autosomal dominant iron load, and tissue iron deposition. So you're basically having the opposite problem where you do not have enough of, basically this IRP is binding really, really strong to the heavy chain mRNA, and that is preventing enough heavy chain ferritin from being synthesized. And if you don't have enough heavy chain ferritin being synthesized, you end up resulting in too much labile and excess iron in the cells. And one of the negative consequences of that is that you're going to have iron overload. You have way too much of this really caustic, I shouldn't say caustic, but a highly reactive ion that is in your cells, and you just aren't able to sequester it. So I guess this is where the double-edged sword is coming from. Sometimes proteins that you synthesize, if they're in too high concentrations, they can precipitate or aggregate, and that can cause disease. But if you do not have enough proteins that are supposed to be fulfilling their jobs in protecting you from these chemicals, then what ends up happening is the chemicals then start damaging you and then causing pathologies. So remember, it's everything in biology is self-regulating, trying to regulate itself, but if it ever gets pushed over the edge, then you can end up having a disease in either direction. So I find, yeah, that is actually very interesting. So in addition to mutation-induced alterations in H and L ferritin, mutation-induced alterations in the expression of other IRE-modulated proteins can also be disease-relevant. Mutations in the 5' untranslated region downstream of the IRE motif of ferroportin mRNA, this have been detected in patients affected by hemochromatosis with iron overload. And hemochromatosis, I'm not exactly familiar, and it's basically just too much iron building up in your body. So ferroportin is the major and sole iron exporter that transports iron ions out of cells. The mutation in the 5' untranslated region of the ferroportin transcript may alter the IRE-IRP interactions, inhibiting which inhibit translation of ferroportin and impairs the exportation of iron from cells. And this is inducing iron overload. So if your cells are not able to pump iron out of them efficiently, and that requires having enough of these iron transporters on the surfaces of the cells, then the iron can build up within the cell and cause disease that way. So on the other hand, mutations in the coding area of Ire modulated genes have also been found to be relevant to human disease. The mutations of ferritin that lead to the impaired capacity of ferritin to retain iron within its core have been linked to Parkinson's disease as well as neuroferritinopathy, a severe dominantly inherited movement disorder characterized by the deposition of iron and ferritin in the brain, normal or low serum ferritin levels, and highly variable clinical features. So let's take a step back. What is happening? So in this particular case, the mutation of ferritin that lead to the impaired capacity of ferritin to retain iron within its core. This has been linked to Parkinson's disease as well as neuroferritinopathy. So let's go back to this diagram here. So ferritin, this is one of the proteins. These are the proteins that when you have iron in your system, like a lot of irons in your cells, it is going to induce the production of more ferritin to help sequester those labile irons, right? Now, let's say you are unfortunate enough to have the mutation that prevents ferritin from binding iron properly. So in this particular case, let's run through this. You have, let's say this is the ferritin gene. You then have a lot of iron. You ate a hamburger and you have a lot of iron in your cells right now. This iron will bind to the iron regulatory proteins, which then kick it off of the mRNA. And then what happens is this mRNA is then going to encode for ferritin. So what ferritin will do is ferritin, after it's synthesized, will then take some of these irons off of the iron regulatory protein and sequester them inside their iron core. And by sequestering the iron into the ferritin, this iron response protein or the iron regulatory protein is now free again to then bind to the iron response elements and then inhibit the production of more ferritin. Okay, so that would be a negative feedback loop. Now, let's say you are unfortunate enough that the ferritin does not bind to the iron as strongly as it should. Well, as a result, this iron regulatory protein, there will still be too much iron left in the cell. So you will not be able to turn off the production of more ferritin. And that ferritin is not going to be as good. So you are hyperproducing this ferritin that is an inferior product, trying to make up for the fact that it cannot sequester iron as well. So you'll produce way too much of it. So then you are basically getting the worst of both worlds. It's no longer a double-edged sword. It's just stabbing you. So you will have too much iron in your cells so that you will get iron overload, but then you will also be overproducing all of the proteins that are going to be upregulated by the presence of iron. All of those are going to be shooting through the roof as well, which is why you're probably seeing effects such as or pathologies such as Parkinson's disease arise from this particular mutation. So yeah, that would be an example of your self-regulation or your homeostasis falling out of whack as a result of a mutation. So as well as neuroferritinopathy. So then this neuroferritinopathy is a deposition of iron and ferritin. So it is both iron and ferritin that you're seeing in extremely high concentrations being deposited because there's just too much of it. So you have too much of this and you have normal or low serum ferritin levels. So and highly variable clinical features. So it's low in the serum for some reason, and I would not be able to tell you exactly. Okay, that just know that that's some of the pathologies. That's a little hard to wrap my mind around. Anyways, mutations at the C-terminus of the L-ferritin impair its stability and decrease its capacity to interact with iron, validated by the transgenic mouse models expressing mutant L-ferritin. So other disease-relevant coding region mutations have been reported in other IRE-encoded genes, including the erythroid 5 aminolevulinic synthase, or ELS, linked to the sideroblastic anemia. So What is sidero? Sidero means iron, and sideroblastic, I would, so that would be a blood disorder in which the body has enough iron but unable to use it to make hemoglobin, which carries oxygen. So that would be having way too little hemoglobin, which is why it's called an anemia. Um, so yeah, if you don't have enough of this 5-aminolevulinic synthase, then you can get this type of anemia, and if you have a mutation in the ferroportin, you can get the hereditary hemochromatosis, and if you have a mutation in the divalent metal transporter 1, you can have another disease. So yeah, there's a lot of diseases that can be the result of mutations in the IRE encoding genes, not necessarily in the iron response element, though. So potential novel therapies using IRE inhibitors for human neurodegenerative diseases. So the IRP-IRE signaling pathway has been implicated in the modulation of amyloid precursor protein and alpha-synuclein translation, which is important in the neurodegeneration of Parkinson's disease and Alzheimer's disease. Therefore, the identification of small molecular iron response element chemical inhibitors to reduce APP and alpha-synuclein levels and alleviate protein aggregation have therapeutic significance to human neurodegenerative diseases. So this is a target. So this is frequently what happens in pharmaceutical design is we find these metabolic pathways, or we find these cellular signaling pathways, and we look for these positive and negative feedback loops. Just some form of signaling is being screwed up here, right? And then we can develop pharmaceuticals that we know will, or what I should say, that basically will modulate the activities of those cellular signals or those chemical feedback loops. And as a result, we can screen a bunch of different chemicals to see which one elicits the best response. So, in principle, identified chemical IRE inhibitors can decrease ferritin and transferrin expression to alleviate the excess iron accumulation in Alzheimer's disease or Parkinson's disease. So, if you decrease transferrin and if you decrease ferritin, you end up having less iron accumulation in the Alzheimer's disease or Parkinson's disease brains. So if you block transferrin, that's going to block your ability to even absorb enough iron from your diet, if I am remembering correctly. And I believe ferritin might also be in the brush border cells, and it's also in the blood. So regardless, if you downregulate all of this, you will probably not be absorbing iron the same way. So furthermore, therapeutic iron response element inhibitors can downregulate APP and alpha-synuclein protein translation and inhibit protein aggregation, and this can promote neuronal survival. Therefore, potent non-toxic pre-drug IRE inhibitors with excellent BBB, blood-brain barrier penetrating capacity should have high therapeutic significance in neurodegenerative diseases. And so far, some promising drug candidates of IRE inhibitors have been identified and are being tested in different human clinical trials for AD and PD. Now, one of the things that I am a little bit worried about is that I do know that this transferrin, this plays a very important role in the differentiation of the oligodendroglial cells. So you have glial cells, you basically have stem cells in your brain, cells with multipotent activity, and those will differentiate into the oligodendroglial progenitor cells and then differentiate further into the oligodendroglial cells. Now, the oligodendroglial cells have a bunch of branches and arms on them, and it's a very complex structure. And what this does is those arms or those branches will go out and repair the myelin sheath of the nerves in the central nervous system. So if you have damaged neurons, and I should say the myelin sheath, this is the insulator that is over the axons of the nerves, which helps conduct the electrical signal or the electrical impulse through the nerve cells. So as a result, over time, sometimes you can have a lot of damage of these myelin sheaths. For instance, multiple sclerosis would be a prime example of what can happen when you have the lack of myelin sheaths. So this myelin sheath is reproduced or repaired by these oligodendral glial cells. But the differentiation into oligodendral glial cells is mediated by adequate supplies of transferrin. Okay, so what worries me is that if we downregulate transferrin too much, that might have some unintended negative side consequences with trying to repair the brains of people with Parkinson's disease or Alzheimer's disease. So that's just one of the things that I'm a little concerned about. So with that, let's continue. So far, some promising drug candidates of IRE inhibitors have been identified and are being tested in different human clinical trials for AD and PD. So in one recent study, 13 potent APP translation blockers that act selectively towards the uniquely configured IRE RNA stem loop in the 5' untranslated region of amyloid precursor protein. So remember, these iron response elements can be significantly different between different mRNAs or between different genes, right? So this APP, if you have a drug that can target this APP's iron response element, hopefully it will not bleed too much into these others. Or let's say you want to develop a drug that can affect multiple genes, but you only want it to affect the genes that you want to attack. And by selecting the genes that you want to attack, you can have a more targeted approach to your pharmaceutical design, and hopefully less side effects and less negative consequences are going to result from that drug if it's targeted. So with that, let's go to the APP mRNA. We basically looked at a lot of compounds of a chemical library at Harvard. So one recent study, basically they looked at 110,000 different compounds, and 13 of them seem to have the potent ability to block translation of amyloid precursor protein by binding to its specific iron response element RNA stem loop. And it did not affect, or it was specific at the APP versus other iron response elements. So some of these chemicals were able to selectively reduce neural APP production in this SHSY5Y cells, so this would just be a cell line, without affecting cell viability or the levels of alpha-synuclein and ferritin. So this is good if you only want to get rid of the amyloid precursor protein, but not affect the alpha-synuclein nor the ferritin. You could use some of these chemicals. So some of the 13 were able to explicitly target amyloid precursor protein. So in this study, the identified APP blocker 9, or JTR009, which is a benzimidazole, which that's just a chemical family. This is found to be superior to the other APP blockers in its ability to reduce the production of toxic amyloid beta in SHSY5Y neuronal cells. So JTR009 is thought to directly interact with the IRE in the 5' untranslated region of APP mRNA and constitutively repress amyloid precursor protein translation. Furthermore, pifithrin alpha, or PTFA, an anti-stroke agent and a p53 inhibitor, and p53, just know that this is one of the cellular signaling proteins in your cells. This was also found to have selective APP translation inhibition capacity. However, JTR009 was able to selectively inhibit amyloid precursor protein expression, while PTF-alpha could cause a dose-dependent downregulation of APP, alpha-synuclein, and actin proteins in cells. So, so PTF-alpha is affecting more different proteins, not just amyloid precursor protein. So furthermore, the potency of JTR009 to inhibit the amyloid precursor protein 5 untranslated region conferred translation was greater than that of positive fenserine, a well-recognized and tolerated IRE inhibitor with both APP and alpha-synuclein translation inhibitory capacity. So they're saying this JTR009, which is specific to APP, was greater, like it had more ability to inhibit translation than this less sensitive posifen, which is positive phenserine, which is probably where the name posifen comes from. So, posifen is a small molecule drug derived from calabar beans and it is a phenylcarbamoyl analog of positive phytostigmine. Okay, so identified from a natural product NP chemical library or nature product chemical library, posifen is found to inhibit both APP and alpha-synuclein protein translation. So furthermore, so this is also very common where we will scan a bunch of natural compounds uh, in nature, right? And we scan them and we find ones that end up doing having one of the effects that we're looking for. And once we find that effect, or once we find that particular drug type found in nature, some biomolecule that is able to have some of the effect that we're looking for, we can then take that same molecule and then we can do a bunch of chemical modifications on it. And by doing those modifications, we can test all of those different other chemicals and we can find a chemical that is even better at eliciting that effect that you cannot find in the original, or that is far superior in the natural product. So furthermore, the inhibitory effects of posifen are potent and not toxic. So however, phenserine, its enantiomer with cholinesterase inhibitory capacity, has less inhibitory potency than posifen, as well as minor toxicity. So don't worry too much about that. So another study demonstrated that posifen and its metabolites, as well as phenserine, possess neuroprotective/neurotropic capacities at concentrations of clinical relevance. So let's look up posifen Wikipedia. So we don't have that, but is a phenyl? Let's look up phygostigmine. Oh, so it's toxic normally. Interesting. So, oh, that's another thing that we do in these chemical trials, is that sometimes a natural component can have toxic effects, and by doing some chemical modifications, we can sometimes find chemicals that have far less toxicity associated with them. Okay, well, let's continue. So, however, so all compounds are found to potentially inhibit the protein translation of APP and alpha-synuclein in neuronal cells. Therefore, posifen and its metabolites, as well as phenserine, may be drug candidates for Alzheimer's disease and Parkinson's disease. So the posifen-induced inhibition of Ire has been validated by various in vivo, in life, and in vitro in test tube studies. Recent phase 1 human clinical trials and proof-of-concept studies in subjects with mild cognitive impairment, or MCI, have demonstrated that the brain entry and capacity of posifen to lower APP production in subjects as assessed in cerebrospinal fluid by as much as 50%. Yeah, so if you can lower the APP production by as much as 50%, you could seriously reduce the amount of plaque that is forming. So currently, phenserine is under phase 3 clinical trials for Alzheimer's disease, while posifen is in a phase 2 human clinical trial for AD and PD. So other potential chemical Ire inhibitors have also been reported in a pilot study. Paroxetine, which is a serotonin reuptake inhibitor, N-acetylcysteine, which is an antioxidant in iron 2+ chelator, or NAC. So that's also important to note, that this N-acetylcysteine is able to bind to iron, and it's also an antioxidant. Anyways, these were employed to assess their anti-amyloid capacity in the transgenic TGCRND8 Alzheimer's disease mouse model. So this is just the name of it, and it's a mouse model that gets Alzheimer's disease pretty much guaranteed. And the levels of amyloid beta peptide were found to be reduced after the exposure of mice to paroxetine, N-acetylcysteine, and erythromycin. Interesting. Okay. So other studies have verified that paroxetine can modulate APP expression through its actions on the 5' untranslated region of the amyloid precursor protein transcript. Therefore, paroxetine may be a chemical IRE inhibitor that can modulate APP expression. Another high-throughput screening study to search for novel, non-toxic, and selective small molecule inhibitors of alpha-synuclein expression have been performed. From 303,000 screened compounds, one compound, ML150, displays very potent inhibition of alpha-synuclein expression. ML150 specifically reduces alpha-synuclein translation and likely acts through an interaction with an inhibition of IRE of the alpha-synuclein transcript. So let's see if ML150 is here. So yeah, supposedly this compound is able to interact directly with the iron response element of alpha-synuclein. Anyways, these studies suggest that ML150 is a novel IRE inhibitor that specifically modulates alpha-synuclein expression. The detailed molecular structure of the identified IRE chemical inhibitors, as well as their potential relevance to therapies against human neurodegenerative diseases, are illustrated and summarized in Figure 3. So here's Figure 3. Just know that these chemicals have the ability to target the translation of particular proteins that have these iron response elements on them. So some of them I would presume would be able to, I think all of these are downregulators, but theoretically you could find upregulators as well. Anyways, the pathologies and therapeutic targets in the iron IRP-IRE signaling pathways for Alzheimer's disease and Parkinson's disease. So there has been an accumulation of evidence regarding the pathological iron accumulation in Alzheimer's disease and Parkinson's disease brains. Excess iron deposits in the brain can induce oxidative stress, leading to protein aggregation and neuron vulnerability. Furthermore, an increased iron levels can lead to upregulation of translation of APP and alpha-synuclein, which are two key proteins vital to neuron degeneration in Alzheimer's disease and Parkinson's disease. Therefore, blood-brain barrier penetrating iron chelators should have therapeutic significance for Alzheimer's disease and Parkinson's disease patients. And the administration of desferoxamine, or DFO, which is an iron chelator. So desferoxamine, if you have iron overload, it is a drug that can be given to you to help sequester that iron, so it's easier for you to remove it from your system. The administration of desferoxamine can slow dementia in Alzheimer's disease patients. However, DFO is unstable and it has poor BBB permeability. So cleoquinol, which is an iron, copper, and zinc chelator with BBB penetrating capacity, this can clinically alleviate cognitive loss and decrease plasma amyloid beta levels in Alzheimer's disease patients. However, cleoquinol has been identified to be associated with myelinopathies. A second-generation alpha-hydroxyquinoline analog, metal chaperone, PBT2, this was found to be superior to cleoquinol with little adverse events. So what is PBT2? It's a zinc ionophore and an experimental drug candidate, and it is and it's intended to be a successor to cleoquinol, which is a potential treatment to Alzheimer's disease and Huntington's disease. So interesting. So PB2 can improve cognitive function, improve cognition in aged APP C50. Okay, these transgenic mice, right, that have they have mutations that probably cause excess production of APP, with the effects associated with the decreased interstitial amyloid beta levels. So furthermore, a phase 2 trial with PBT2 in 78 AD patients showed a reduction in amyloid beta levels in cerebrospinal fluid and enhanced performance metrics. So other iron chelators, including VK28, HLA20, and M30, can both suppress APP expression and lower the amyloid beta levels in the brain. So basically what's happening, these chelators are coming in, they're binding to iron, and by binding to iron, the iron is not able to bind to the iron regulatory protein. I'm presuming that is the main case. And by doing that, you're having a large downregulation of the amyloid precursor protein because remember, amyloid precursor protein is being upregulated in the presence of high levels of iron. One of the reasons for this is because it is important in helping get the iron out of the cells. So these results suggest that iron chelators induce downregulation of APP and amyloid beta levels through the iron regulatory protein inhibition of APP translation. So iron chelating agents have also been validated to be effective in Parkinson's disease therapy. A natural iron chelator, phytic acid, this has been found to protect dopaminergic neurons in an in vitro Parkinson's disease model. So phytic acid, you might have heard about this a lot. So phytic acid, I believe it's inositol with 6 phosphate groups on it, but this has frequently been talked about as being an anti-nutrient. It prevents nutrients from being absorbed properly. So let's go to the Wikipedia. Yeah, so it's a 6-fold dihydrogen phosphate ester of inositol. And so this is basically the molecule. It's just a lot of phosphate groups bound to an inositol moiety. Now, this is a strong binding affinity to dietary minerals, calcium, iron, and zinc, which is inhibiting their absorption, right? But this phytic acid has been found to protect these dopaminergic neurons in an in vitro Parkinson's disease model. Now, granted, that's when the phytic acid's in the actual, like, touching the actual cells of the target neurons. Granted, when you are eating phytic acid, you don't really, I don't believe you absorb a lot of it. I think it's just such a large molecule, it can't be absorbed very readily. And then also it's binding these metals, so you're basically passing it through. So regardless, furthermore, iron chelators, including D-ferroxyrox, diferroprone, and diferoxamine, all of these, they can all significantly attenuate or lower the loss of dopaminergic neurons in various in vivo Parkinson's disease models. So a lot of these iron chelators are very beneficial at protecting your neurons. So, detailed future studies are expected to further establish clinical efficacy and safety of iron chelating strategies in Alzheimer's disease and Parkinson's disease therapies. So, the intactness of the Ire motif is vital to the function of the IRP-Ire signaling pathway. Conceivably, point mutations in key nucleotides of the Ire motif of target mRNAs can disturb the Ire stem loop structure and abrogate or negatively influence the interaction between the IRP and the IRE, which can be a pathogenic factor contributing to neuron degeneration in Alzheimer's disease and Parkinson's disease. So this point mutation of the Ire motif of heavy chain ferritin gene can lead to an inherited human disease associated with perturbed iron metabolism or iron overload. You just have too much iron and it's not being sequestered by the heavy chain of the ferritin. Anyways, studying whether mutations of the iron regulatory elements of heavy ferritin and other iron metabolism relevant genes are susceptibility factors for Parkinson's disease and Alzheimer's disease will be intriguing. So furthermore, point mutations in the Ire motif of APP and alpha-synuclein gene can be a pathogenic factor in Alzheimer's disease and Parkinson's disease, which is a well-described single nucleotide polymorphism associated with Alzheimer's disease risk exists in the 5' untranslated region of the APP gene. The pathogenic single nucleotide polymorphism in the 5' untranslated region of APP is hypothesized to disturb the Ire motif, leading to an increased AD risk. Therefore, more attention should be paid to potential disease-relevant mutations of single nucleotide polymorphisms in the Ire motifs of the APP and alpha-synuclein genes. So, you know what? I'm going to skip the rest of this. There's only 2 paragraphs, and we've basically been repeating ourselves quite a lot. So that's what they tell you when you do paper writing, and when you write anything, you really need to repeat your point over and over again. By repeating your point over and over again, you make sure that your point, your thesis, sticks in someone's brain. But then also you're trying to say something give more details, say the same thing again. That way, when someone is rereading that original fact that you, or that original thesis, they're able to recomprehend that thesis with the new details that you've given, and then you give more details, and then say your thesis again, give more details, say your thesis again, and that's a really good or really effective strategy at getting a thesis stuck in people's heads, and they can understand your thought process of why you approached or why you reached that um, your thesis. So I think that's good. So with that, I'm going to head on out. Just things that I want to note is that these iron chelators might play an extremely beneficial role in preventing neurodegenerative diseases, because screwed up iron regulation is, I mean, it's one of the most important minerals in the body, right? Your iron is one of the highest concentrations of a metal ion in your body, right? So as a result, it is extremely important that it is, any dysregulation in it is going to severely hamper your ability to be have chemical homeostasis in your cells. So yeah, this is why, I mean, this is why I drink green tea all the time, because it is a known iron chelator, and it can help prevent the reactive oxygen species production of iron. So even if you have the iron floating around, it's not going to produce as many free radicals if it's bound to the EGCG, which is found in green tea. So there are a lot of different strategies that can be employed, and strategies that you as a consumer, and you as just a general layperson, you can take steps in order to benefit your long-term neurological health, just by purchasing or consuming particular products versus others. So I think with that, I've over what would the word be? I've overextended my stay, right? So thanks for watching, and if you have any questions, please leave them down in the comments below, and hopefully I will get to them if it's not spam. So thanks for watching, and hopefully I'll see you later.